Okay, can everybody hear me all right and see my screen?
Yes, we can.
Okay. Okay, so as Catherine said, my name is Brendan Mormile, and I'm a PhD candidate here at Texas A&M. And today I'm going to be talking to you about primer design for the polymerase chain reaction. So to begin with, I want to first go over some of the basic steps that are associated with a PCR. Whenever you perform a standard PCR, you're likely going to be using a double-stranded DNA template from either genomic DNA or plasmid DNA. Now, the first step is to denature this double-stranded DNA by breaking the hydrogen bonds that link them together. Now, you're going to do this by increasing the heat to about 98 degrees Celsius. This will produce two single-stranded DNA molecules, which will expose the areas that will allow your primers to bind. The temperature is decreased from 98 to approximately 50 to 60 degrees Celsius. This is called the annealing temperature. This is the temperature where your primers will bind to their complementary regions within the single-stranded template. Now, the temperature will then be increased, and the temperature will be dependent on whichever polymerase that you're using. In this example, they're using the TAC polymerase, which is optimal at 72 degrees Celsius. This is the temperature which the polymerase will then create the DNA elongation strand. And then these templates will then be shuttled back to the beginning, denatured, and the entire process will repeat itself. Now, what exactly are primers? Primers are very simplistic. They are short, single-stranded DNA fragments that bind upstream and downstream of the DNA region being amplified. They are very often used in pairs called either the forward primer or the reverse primer. Now, when a double-stranded DNA template is denatured, it will produce two single-stranded DNA templates deemed either the 5' to 3' template or the 3' to 5' template. The forward primer will always bind to the...
Sorry, Brendan. It appears that some of them cannot see your screen. Can you all see the screen that Brendan is sharing?
Yes.
Okay. Sorry, you can go ahead. Yeah.
Okay. Yeah, if there's any further problems, just let me know and I'll pause and correct it. So your primers will always bind to the 3' end of whichever template they correspond to. So your forward primer will always bind to the 3' to 5' strand, and your reverse primer will always bind to the 5' to 3' strand. However, both primers always bind to whichever 3' end is present. Now, primers should also be complementary to their corresponding sequence on the template. As you see here, the A's match the T's and the G's match the C's. However, primers can be tolerant to a certain number of mismatches. Mismatches are predominantly tolerated within the 5' end of your primer. The 3' end, however, must be complementary for elongation to occur. In this example right here, this A must be an A in order to match the T. If this is a T, a C, or a G, this will have what's called an overhang. This will not be flush to your DNA template, and the polymerase will not be able to elongate your strand. I'm going to show you an example of this a little bit later on. And the same thing goes for your forward primer. This must be a T. Now, I want to go over some of the properties that are associated with the most effective primers. Now, your primer length should ideally be between 18 and 24 bases long. Anything less than 18 bases, you're going to have a increased likelihood for off-target binding. Remember, with genomic DNA, you can be dealing with billions of different base pairs, and if you have less than 18, you're going to be opening up the door for off-target effects, non-specific bindings to some alternative location within the genome. If you have more than 24 bases, then you're going to have a slower hybridization rate. There's going to be more bases that need to bind to more locations, and this is going to slow down your reaction. Now, you also want to have a GC content of approximately 40 to 60%. All this means is the number of G's or C's present within your primer. In this example right here, we have a 20 base primer, and 10 of those bases are either G's or C's. This gives us a GC content of 50%. Now, a GC bond has 3 hydrogen bonds compared to an A and a T, which only has 2 hydrogen bonds. Now, the GC 3 hydrogen bonds will enhance the target stability, and it will make for a tighter binding primer to your template. Now, similar to GC content, it's also ideal to have a GC clamp. And all this means is that your primer begins and ends with a G or a C. In this example, we have a G or C on the 5' end and a G or C on the 3' end. This is more important to have a GC clamp on the 3' end because you want your 3' region to be very tightly bound. So in order for the primer to perform elongation. It's not as important on the 5' end, but you could include it as well. The melting temperature, this is a critical factor within primer design. Okay, you want your melting temperature to be between 50 and 60 degrees Celsius. Now your melting temperature is the temperature where half of your primers will disassociate with the DNA and the other half will bind. Now your primer pair, your forward and your reverse, should have a similar melting temperature, ideally within 5 degrees of each other. Now the melting temperature is important because it will directly influence your annealing temperature. Now, whenever you perform a PCR, you typically begin with an annealing temperature that's 3 to 5 degrees below your melting temperature. So, for example, if I had a primer pair that had a melting temperature of 60 degrees Celsius, then my annealing temperature would ideally be about 55. So if my melting temperature is 60, half my primers will bind at 60. At 55, more than half my primers will bind to the DNA template. Now, primer pairs should also not have any regions of complementarity. In this example, my forward and my reverse primer both have areas of complementarity within their 3' region. This can cause what's known as a primer dimer, where your primers will associate with themselves instead of your DNA template. And it's similar how you don't want your primers to bind to each other, you don't want your primers to bind to itself either. So you do not want any internal secondary structures. In this example, I have a primer which has 4 A's and 4 T's on the 5' and 3' ends. This could potentially fold in on itself and create what's called a hairpin structure, which is going to hinder its recognition to the target site. Okay, now these are the parameters that are associated with optimal primers. Depending on whatever your experiment is that you're running a PCR, you can sometimes either bend the rules on this, and some of these you can completely break. And I'm going to go over some examples of how you can do that. So before we go into some more complex primer design methods, I'm going to show you how to very simplistically manually design a primer set. Okay, so I'm going to be showing you how to design a primer that can amplify this DNA fragment. Okay, so first we are going to denature our DNA template. And I'm going to switch over to the pen. Okay, now we're going to first start with our forward primer. Now remember, primers will always bind to the 3' end of your DNA, single-stranded DNA template. Okay, now we're just going to very simply write the corresponding complementary nucleotides, which is going to be an A, T, A, G, C, G, T, A, C, G, T, C, C, T, G, A, C, C, T, T. Now this is 20 nucleotides long, and I end on a T. Now I'm just going to extend this 1 nucleotide over to a G that's 21 bases long. Now we end on a GC clamp. So this is annotated as 5' to 3'. So this is where my polymerase will bind to. and elongation will occur in this direction. Okay, so this is my forward primer, and this is all set. Now, your reverse primer is slightly more complicated, but it's relatively similar. So this will be my reverse primer. Again, I'm going to start at the 3' end, and I'm going to do 20 nucleotide primer. So A, C, G, T, A, C, G, A, C, T, C, A, G, G, T, C, A, G, G, A, C. I think that's about 20. And this ends on a GC clamp. This is my 5' region. This is my 3' region. Our polymerase will bind right here, and elongation will occur in this direction. Now, one thing I want to point out is that you always write your primer sequence in the 5' to 3' direction. So this primer, the forward primer right here, is annotated as AT, AGC, so on. The reverse primer is annotated in the ACG formation. So just to clarify, this is what my final primer set would look like. Everything is always written. The forward primer is written in the 5' orientation. The reverse is also written in the 5' orientation. Okay? Now, let's look at some more complicated design tools used for primer design. Okay, can everybody still see my screen?
Yes.
Okay, so the first thing we're going to be looking at is the NCBI website. Now, the NCBI website is a great database that can host most of the annotated genome sequencing and can be used for various degrees of molecular tools. So the gene I'm going to be looking at to amplify with my PCR is going to be CLOUT1. So I'm going to select gene from the drop-down menu, type in CLOUT1. Now, CLOUT1 is a gene that's responsible for chloroplast development. There's some form of CLOUT1 found in pretty much every single plant species that you're going to be working with. So this is a great control gene that we typically use. The primary plant that I work with is Giusepium hirsutum, or upland cotton. So this is the gene I'm going to be showing you as an example. So if you were to perform any kind of genome sequencing or genotyping, and you just want to detect the presence or the absence of a gene, NCBI is a great tool to use. So I'm going to select CLOUT1. This is going to give me the full name of the gene, what it's responsible for, a brief description. So it's going to show me where on the genome it's located. It's located on chromosome 10, and there is a 10 exon count. And down here, it's going to give me a nice illustration of the entire gene with all 10 of my exons separated by the introns. Now remember, if I'm just looking for the presence or absence of a particular gene, this is a fine tool to use. So I'm just going to right-click, select range, and I'm just going to kind of non-discriminatory select the entire gene. Okay, now I'm going to do a primer blast of my selection. Now right here, it's going to give me a few basic parameters that I need to plug in order to optimize my search. The ascension number, this is just CLOUT1. This is their code for their database within NCBI. This is the range that I selected. The forward primer will bind somewhere around this location on the chromosome, while the reverse will bind somewhere upon this location. Now, a PCR product size, a PCR can basically amplify a region roughly at a minimum of about 50 and a maximum of about 10,000 bases long. I don't really care about how big my PCR product is because I'm just looking for the presence or the absence of a band when I run it on my agarose gel afterwards, the presence, the absence of a PCR product. The number of primers returned, this is set at default at 10. If you increase this number to how many results they're going to show, it's going to slow down your the loading of your results page. I'll just keep this at 10 right now. The melting temperature, as I said earlier, we'll keep this at a minimum of 50, a maximum of 60, with our optimal temperature being about 55, and a difference of about 5 degrees between the two, the forward and the reverse primer. The exon intron selection, this is if you were to design primers primarily for an RT PCR. This is a little bit different. This is basically looking at the messenger RNA after the intron's been spliced out. This will select primers that only binds to 2 separate exons. So after the 2 separate exons have been spliced together, you're gonna have a primer that binds to the junction. This is a little bit more complicated. I'm not gonna go into too much detail about this. However, the primer pair specificity checking parameters are somewhat important, especially for genotyping. So this is going to look for alternative locations within my host organism, the Giuseppe hirsutum in this example, where your primers could potentially bind to. And it's going to eliminate primers that can bind to more than one location within your genome. So the results page are going to give me primers that are very unique and very specific just for the gene of interest that I'm looking for, CLOUT1. Okay, now let's take a look at our primer selection. Now, depending on your internet connectivity, this can take several minutes for your primers to load. So I've already performed a primer blast and I have the results page loaded right here, but your results page will look something like this. So right now it's giving me my top 10 choices of primers that will amplify various regions within the gene. And if I scroll down, it's going to show me my forward and reverse primer shown in the 5' to 3' annotation. It's also going to show me the length of my PCR, or the length of the primers, and the length of my PCR product. It's also going to show me my melting time, my GC content, the number of self-complementarity. This is the likelihood that your primers will bind to each other, and your self-complementarity 3' is your likelihood that your primer will bind to itself. The higher the number, the higher the likelihood that you're going to get some sort of secondary binding. However, these numbers are okay for the time being. Okay, so this is how you would design primers using the NCBI website. Now, let's say I wanted to give a little bit more specificity as to where my primers will bind to. So I'm going to come up to the database at NCBI. I'm going to locate my gene ID number. I'm just going to copy this. Now we're going to take a look at what's called Benchling. Benchling is a free-to-use online network. You need an account, but the account's free to obtain. And this is a fantastic platform to use any kind of molecular biology, PCR, CRISPR, anything like that. So I'm just going to scroll over to Create. I'm going to go to DNA sequence, new DNA sequence. I'm going to search an external database for my sequence. I'm going to be using the NCBI extensions. I'm going to copy the gene ID paste the gene ID that I just copied, and I'm going to perform a search. Now, this is going to show me all the information that NCBI has. This is the gene of interest, CLOUT1. I'm going to import this sequence. And it's going to give me a nice illustration of my entire gene sequence. It's going to show me all the exons, and it's going to even show me my amino acids that they correspond to. Switch over to linear. Now, let's say hypothetically, I wanted to know if exon 6 is present in this gene, okay? So that means I only want primers that will amplify roughly from this area. So what I can do here is I can design primers that will amplify this region. I could run my PCR product on agarose gel, and I could look for a band that is approximately 274 bases shorter than my other PCR products. This will indicate the absence of a exon 6. So now I'm going to look for primers that will amplify this region. So I'm just going to drag my cursor, and I'm going to select this region that includes exon 5, 6, and 7. Now I'm going to right click and run primer 3. Okay, so similar to NCBI, I'm just going to use my selected region. I'll keep my GC content between 40 and 60, my melting temperature between 50 and 60, optimal at 55. PCR band length will be between 18 and 24, and it's going to give me 1 GC clamp on my 3' end. Okay. The amplified region can be between 500 to 4,000. This is within my selected region. I'm not going to switch anything here. You can generate how many different results will load. We'll keep this just at 5 for simplicity. And there are a couple other parameters that you could include if you wanted to, but these are a little bit more advanced, and I'm going to keep it kind of simple for today. So right now, let's generate our primers. And right here on our reverse page, it's going to show me all the primers, or our top 5 primer selection. It's a GC percentage, melting temperature, location as to where they bind, number of nucleotides within the primer, and the projected the PCR product size. And my primer annotated in the 5' to 3' orientation. And I can even look at my map. If I select all of these, I can see exactly where they bind on my gene. So if I wanted to, I can just go right here. I can select whichever one I'm looking for, and I can order this from whatever ordering platform that you're using. Okay, so that's how you perform some sort of a genome typing. Now, let's say I wanted to do a PCR in order to clone an entire gene out of a genome and clone it into a plasmid for whatever reason that you might be looking for. And we're going to be looking at the restriction cloning technique here. Okay, so I'm going to go back to NCBI. Remember, I use NCBI for many, many different things. It's a great platform if you want to find a gene sequence. All you need to do is go to FASTA. And this is going to show me my sequencing of CLOUT1. So I'm going to take the sequencing information and copy it. Now I'm going to switch over to snapgene Viewer. Snapgene Viewer is the free to use site. There's also snapgene in general, which is pay to use. Snapgene Viewer is free, and again, you can just create an account, no problem. So I'm going to come over here. I'm going to paste my sequencing information. I'll call this file Giuseppe Hirsutum CLOUT1. Okay, similar to Benchling, it's going to give me my sequencing information. Here we go. So I want to amplify this entire gene. So now I need to be more selective than the other 2 examples about where my primers will bind to. So I have to have a primer bind all the way at the beginning. So I'm just going to drag my cursor for about 20 nucleotides, and you can see up top in the purple, my melting temperature is 57 to 58 degrees Celsius. So that's perfect. Also, my GC content is 50%. That looks good. So now this is my primer that I will be using. So I'm going to come up top, select primers, add primer. This is going to be my forward primer. Amplification will occur to the right. So I'm going to select top strand, and it's going to give a nice visualization as to where my primer will bind to, and it's going to point to the direction of elongation. Now, if I'm performing a restriction clone and I want to clone this PCR product into some sort of a plasmid, I need to add a restriction site to my 5' end. Okay? So I'm going to come up here to insertions. I'm going to select my 5' end, and I can select whichever restriction enzyme site I want to use, whichever site that I'm going to be cloning into the plasmid. So for this example, I'm just going to select expa, and it's going to give me my restriction site sequence. I'm going to insert. Now, what's good about this is that it will show you the base pairs in red that do not bind to the template. Now remember, I told you that the 5' end is very tolerant of mismatches. So this is okay. If there's mismatches at the end of this primer, it's fine. This will still amplify. Now typically, I also want to add a couple nonsense codons. So I'll just do TGA. Restriction enzymes typically don't like to cut all the way at the end. They typically need a little bit of overhang, a little bit of bulk, about 6 nucleotides extra so that they can cut in the middle, okay? Now I'm going to add this primer to my template. And again, a little bit of this got cut off, that's fine. I can double click it, copy this, and then send this sequence for ordering your primers. Now let's take a look at the reverse primer. For visualization, I'm going to just amplify over here so you guys can see all the overhangs. So I'm going to select 20 nucleotides. And I got a melting temperature of about 54. GC content is 40%. I'm going to go to primers, add primer. Now this time I'm adding, this is the reverse primer. So this will bind to the bottom strand. Okay, and it's going to show me my direction for elongation. Now, I want to point out a very common mistake here. Let's say I wanted to add a restriction site to this, and we'll do X, okay? If I were to make a mistake and I were to add my restriction site to the wrong end, to the 3' end, and this is my final PCR, my final primer, this will not work as a primer. This has an overhang mismatches on the 3' end. This will not bind to my template, and my polymerase will not amplify this region. So this is a mistaken primer. So what I'm going to do, come back here and correct it, and then add my restriction site to my 5' end. And there you go. And add some extra nucleotides. So here we go. So this is a reverse primer. It's got my restriction site on the 5' end. This will amplify the gene and produce roughly a 3,300 base pair product. Now I want to show you one more example about how you can use mismatches. So if I wanted to introduce a point mutation within my gene, I can use this site as well. So let's say hypothetically, I wanted to eliminate this restriction site right here, PSII, okay? It recognizes TTATAA. I want to get rid of this restriction site. And this is also useful if you want to edit any kind of a codon and you want to change an amino acid, okay? So let's say I'm going to select 20 nucleotides, select primer, add primer. Now remember, I'm trying to get rid of this TTAT restriction site. So I want to change this A to a T. So I'm simply going to come up here. I'm going to manually do this. Switch it to a T. Or we could change this to an A. So right now, the restriction site has been removed. This PCR product right here will amplify. This is towards the 5' end, so it can tolerate a mismatch. However, my PCR product will have a A instead of a T, and this will no longer recognize the PSII restriction site. So that is how you design primers using ncbi, benchling, and snapgene. So now I'll open the door for any